And I'm excited to introduce the next speaker. He's our keynote speaker, and it's Douglas Robinson from Novartis. Douglas is the Global Head of Scientific Computing and Consulting at Novartis. And his mission in this team is to enable clinical teams to change how they interact and interpret the data. So without further ado, I will open the floor for Douglas, and I'm excited for his talk.
Thanks so much. Let me share my slides. Okay. All right, hopefully everyone can see my slides okay and can hear me okay?
Yes.
Okay. Fantastic. 1 second. So thanks so much for that kind introduction today. Before I begin, I'd really like to take a moment to thank and congratulate the conference organizers for putting on an amazing conference under such challenging circumstances. I'd like to particularly thank Phil, Daniela, and Harvey for all their support leading up to the conference as well. As stated, I'm Doug Robinson. I have the privilege of leading the scientific computing and consulting team here at Novartis Pharmaceuticals. This is a team of dedicated and highly skilled analysts whose mission it is to interact with and explore our clinical trial data. I'm obligated to share a disclaimer with you. Okay. So the pandemic has made for a surreal existence with many challenging stories in the news, but there are some positive stories as well. For me, one positive is that rather than commuting in and out of Cambridge each day, I get to spend more quality time with my family, particularly my 12-year-old daughter, Samantha. One activity that we've started to do is to take walks around our neighborhood. Here we talk, we look for wildlife, and listen to music. Earlier this summer, my daughter developed a healthy obsession with the Beatles. She's able to name most songs by listening to just a few notes. She knows every album and when it was released. We also look at videos on YouTube. In one video of Hey Jude, actually, in the middle of the song, Paul turns to John and they seem to have a conversation, all without speaking a word and without missing a beat of the song. So I'm going to pause here because as I was developing this presentation, it occurred to me that many of you watching this presentation may not have any idea who the Beatles are. So I'm going to briefly just give you an introduction. So the Beatles were comprised of 4 members, right? So there was John, Paul, George, and Ringo. They hailed from Liverpool, England, and burst onto the music scene in the 1960s. Now, despite their music being 50 to 60 years old, their music and songs still resonate today, as evidenced by my daughter's obsession. Now, we during this time, we took walks, and during one of the walks, my daughter asked me the question that became the title of my talk today. And so she said, so Dad, would John, Paul, George, or Ringo have been famous if it weren't for the Beatles? And this is a difficult question, right? Before I answered, I reflected on John and Paul's seeming psychic ability to communicate and reflected on what teams I've been a part of that have been the most successful. And those are teams that are in complete alignment with one another and information flows freely from one person to another. You know, as analysts in the pharmaceutical industry, we have an obligation to present data that's easily digestible so that our clinicians and clinical team members may make decisions. Unfortunately, it's fair to say that we need a lot of help, and there's a lot of opportunity to improve. Currently, teams pre-plan hundreds of static tables and listings, regardless of of value for our study reports. We generate thousands of formatted output for our submission dossiers and thousands more for our advisory committees. You know, we try to cover all the possibilities, and regardless of that value, this requires heavy resources and time. And unfortunately, many, if not most, are not even referenced in our study reports. You know, then if trials show unexpected results, we must produce even more output. So what are we supposed to do? What we need to do is earn the trust of our collaborators, and we do so by understanding the scientific question. What that means is we have to understand the biology of the disease, the mechanism of action of our drugs, and the technology used to collect the data in our trials. And when we think about that data, we have to identify the sources of uncertainty that could negatively impact the statistical analyses. We have to listen carefully, ask questions, and really think. Chambers once said, "There's no single statistical tool that is as powerful as a well-chosen graph." Effective graphics communicate complex information with ease. They facilitate insight, understanding, and help teams make complex decisions. At Novartis, we formed the Advanced Visual Analytics Initiative to promote graphical thinking and the use of good graphical principles. These graphical principles apply to both static graphics as well as dynamic graphics using R-Shiny to dynamically explore our clinical trial data. So in order to help you learn about and apply these good graphical principles, I present to you the Graphical Principles Cheat Sheet. So this was devised by a group of analysts at Novartis, one of which was Baldur Magnusson, who presented to us yesterday. In addition to the cheat sheet, Mark van der Muller-Brucker et al. Actually wrote a companion tutorial article that goes in and deeply describes that cheat sheet as well. It goes into detail on the 3 points that Baldur Magnusson presented in his presentation. Now, there's a lot of information here in the Graphics Principles cheat sheet, and we don't have time to go over it all, but what I encourage you to do is please go online and download this cheat sheet because it can really benefit you in your next presentation. What we are going to do is just go into this one section right here. So when you think about your graph, you have to carefully consider your question. What is the purpose? And then choose wisely. But you can't just use graphics. You have to combine that graphics with statistical thinking to really inform the decision-making process. Strive for simplicity to maximize the value of your data. So now that you're all graphical experts, it's time for a quiz, okay? So on the next slide, what I've done is I've simulated 3 distributions, okay? And it's your job to determine and answer the questions, are there differences among the simulation runs, okay? When you take a look at this, you might say, okay, well, which graphic am I going to use? All right, it's distributions. I'm going to compare 3 of them. And you might say, box plot, don't let me down. And with that, are you ready? 10 seconds. 3, 2, 1, go. So are there differences among the simulation runs? You know, at first glance, not really. You know, I can't really tell. And if you're anything like me, you're working on multiple projects and you might assume that, you know what, there is no difference and move on to your next project. But should you? Let's go in and explore each one of these distributions individually. So here's the distribution of simulation A. And so I will tell you that I simulated 200 data points from a normal distribution. And honestly, this is exactly what the box plot was meant to display, right? So this actually characterizes the distribution extremely well. Let's go on to simulation B. This one only has 5 data points, right? And what we recognize is that the benefits of the box plot really break down when you only have a low sample size. So although the box plots look similar to one another, I'd be hard pressed to make a conclusion about whether there's a difference or not until I see more information from simulation B. It's hard to say. So let's move on to simulation C. This is the dreaded bimodal distribution, the Achilles heel of box plots. What we see is that the box plot completely obscures the bimodal distribution. And so it's not enough to just use graphics. You have to experiment. And just like Paul and John, you have to allow your data to speak telepathically to its recipient. Let's get back to why things have to change with discussing the potential impact on data monitoring committees. For those of you who may not know, our phase 3 studies are typically designed with one or more interim analyses. And during these interim analyses, the company can make certain decisions based on what they've observed so far in the trial. The trial may be stopped for efficacy or for futility. That's whether the study patients are doing well or not. You can also make other decisions based on the statistical analysis plan, where you can potentially add another dose cohort, you can change the dose, you can add in different dosing regimens, but this all has to be done in a blinded fashion, right? So in order to eliminate bias, what pharma companies do is they hire independent statisticians and clinicians to help make the decisions for us. So Tim Frieda is a professor of biostatistics at the University Medical Center of Göttingen, Germany. He's been the independent statistician on numerous data monitoring committees and has over 15 years of DMC experience and has been on over 50 DMCs. If you're able to download my article, my slides after the talk, this is actually a link to one of his most recent publications on the DMC. So I had a chance to sit down with Tim and ask him about his experiences on DMCs, and here's what he had to say. So he said sometimes the DMC gets thousands of pages of output to review, mostly tables and listings, rarely figures. He said that it's really difficult to see data from different data domains. And in one instance, he said that for a DMC, he received information from a 2019 data cut for a June 2020 DMC meeting. How are you supposed to make decisions on a clinical trial if the data is 6 months old? And then in the middle of DMCs, questions arise, right? Unfortunately, those questions can only be addressed in subsequent DMC meetings. You really want to see the answers, and if nothing, then move on. He also expressed the desire to have the ability to explore ad hoc subgroups. So if DMCs are making such important decisions for us in the pharmaceutical industry, they deserve better. And with that, I'm proud to say that at Novartis, over the summer, we qualified our first Shiny app for DMC use. So earlier this year, Novartis started working with contractors from Rome. And this stemmed from a close collaboration on their interactive safety graphics, which is a sub-team of the ASA Biopharm DIA Safety Working Group. Hopefully all of you caught Jeremy Wildfire's presentation yesterday, where he actually presented version 2 of the Safety Graphics Explorer. Now, personally, I'm incredibly excited about this because it looks like it has some features that we'd like to incorporate as well. What we've done is we've incorporated the version 1 of the Safety Explorer into the Novartis exploration workflow. And then along came COVID-19. So Novartis initiated multiple trials in COVID-19, which used a single DMC. In order to facilitate the review of the safety data, which, by the way, had weekly updates of information, we decided to make a Shiny app. When Tim first saw the Shiny app, he was truly excited about it and enjoyed the experience. He said to me that initially, the DMC members were skeptical, right? They've never seen anything like this before. But over time, they became highly engaged and were able to quickly identify that required information. What he said, one of the things that he liked was that you were able to go from general questions down to specific patients with just a few clicks of the button. That really simplified the data review process. They even came back and requested additional functionality to aid the review. So interestingly, Tim is going to be the independent statistician on another DMC for us next year. And he's expressed strong interest in us being able to present another Shiny application to help with the review. Now, this DMC happens to be fully external, so we can't even have the unblinded information in Novartis. So the question is, how can we host a Shiny app that has the data privacy and data controls based on it so that we can have the DMC be able to use this type of environment? It's something that we're still struggling with, and I'd love to be able to talk with some of you about what your thoughts are. So I'm now going to tell you about a couple of modules that we used to explore the DMC. So here, one of the modules that we did is we used the Adverse Event Explorer. Again, this is based on the Safety Explorer suite from Rowe. This Advent Explorer allows you to characterize the adverse events by system organ class, look at the AE rates both numerically and overall both numerically and by a dot plot. And you can also do a rate comparison with a forest plot. By clicking on the little plus sign here, I hope you guys can see that, you can break down the system organ class into preferred terms. And by clicking on the preferred terms, you actually get the listing of those patients who comprise that adverse event. So again, you can go from summary level down to individual patient information very easily. One of the features I really like is this search box in the upper right-hand corner. You're able to look at the information such as head, which actually searches for headache and tension headache, all at the same time. Another module is the AE event timelines. This is one where it's a swim lane chart which is colored by severity. Here, serious adverse events are easily identified by these black circles, and hovering over any one of these points provides additional information such as the AE term, the start, and end dates. Individual AE charts are able to be clicked by clicking on the individual patient. You can actually drill down and find the specific adverse events for that patient. So beyond the DMC, scientific computing and consulting is supporting a lot of clinical projects, as well as non-clinical projects. You know, to me, I really feel like the clinical projects are the ideal breeding ground for new ideas. What we do is we can see what resonates with our teams, identify those things, and generalize its use across the portfolio. So right now, I'm going to show you a bunch of these examples and show you kind of a general view of what's going on at Novartis. So the first one, we have a longitudinal plot, right? Very standard. We can show means and confidence intervals. This next one was done by Bo Wang, which shows the waterfall plot and box plots. So on a side note, Bo Wang gave that presentation a couple days ago on Meta Shiny. This really seemed to strike a chord with folks. There was a really rich discussion on the chat window during the R and Pharma session. And so what I'd like to say is that we've heard you, and we're going to see what we can do on working on open sourcing that for everyone. Here's an example by Tushan Dong, where it's another swim lane chart where we see the duration of response separated by treatment group. Here, these little points represent intercurrent events for each one of the patients. This one is a Sankey chart. I really like this one. This one actually has been used quite a bit at Novartis. And this one is really good for categorical or ordinal data that's measured multiple times throughout the study. So here what happens is you can actually see if patients start in a certain category. You can see how those categories flow and how they change over the course of the study. So it's really nice to see those summaries and how the patients change their responses over the course of the study. Here's another one. This is called a Lasagna chart, and actually it shows the same information that we saw on the previous chart. And this one here, you have time points on the x-axis, you have patient information on the y, and here we see the different categories in which are colored. And you can see the individual scores over the course of the study. So here on the Sankey chart, you can actually see the different categories, flow and summaries. And the Lasagna chart can show those individual values if you want to drill in and see how individual patients changed their categories over the course of the study. So here we have a situation where we have a really nice Kaplan-Meier curve. This is really the workhorse of the pharmaceutical industry. Here we can show the Kaplan-Meier curve, and it has the number at risk as well as the cumulative number of events as well. What's really nice here is that this is using the Serve Miner package, and what we've done is we've actually implemented it in a time to event module where the next tab actually has the cumulative incidence rates as well as the cumulative hazard functions as well. Another benefit here is that you can actually show both the overall effect and then drill into different subgroups as well, so you can really dig in and explore that time to event information. So you can't have the Kaplan-Meier curve without having a really nice forest plot as well. So this one was done by Shu and was generalized by my summer intern Flavio Lombardo and myself. Initially, it was working for just the time to event hazard ratio information. What we've done is we've generalized it to work for logistic regression to show the odds ratios and confidence intervals. We've even have it working for binomial confidence intervals as well. So it's really quite flexible. Here's one that's an animation, right? And so animations have become very popular at Novartis. This one was actually done by Xiao Ni, who's now at Sarepta. And this one is for composite endpoints. Essentially here, what happens is that a composite endpoint is devised by many different components, and you don't know how each one of the components is influencing the overall composite endpoint. And by looking at this in an animation, you can actually see how that composite endpoint changes and how the different components compare to one another and see which one is influencing that overall composite score. And there's something about the animation that allows you to really understand the evolution of that composite measure over the course of the study. Here's another one that was done by Jahangir Alam and Kaustov Nanda, which shows a day in the life of a patient in a cardiovascular trial. And so here what we see is the patient journey. Here patients generally start at home, and these circles represent the individual patients, right? And so if you press play, you can actually see the dots go to the hospital, back home, maybe to the hospital, to the ICU, to the hospital, to the home. You can see that whole journey there of the patient. And eventually they actually end up, some of them end up in the final state of death. And so it's really nice to see how the patients evolve over the course of that study. This next one is by Mustafa Larboui and David Grandjean. Hopefully some of you caught David Grandjean's workshop last week on R interfaces. So Mustafa built a custom database, and David really built a really nice set of interface, visual interface that can actually do business analytics. Here we can look at the different headcount information and how it's distributed across the different therapeutic areas at Novartis. I will say that David Hall recently joined Novartis, and he's actually contributed really nicely to this graphic as well. The next one and the last one that I'm going to present is the probability of success. So Mustafa Larboui actually worked with our statistical methods team. And what they've done is they've been able to create a nice interface into a very complex statistical methodology to compute the probability of success. By having this really nice interface, what they're able to do is open up this methodology to the entire group, right? And so now it's being applied across the portfolio. And so we can get really good probability of success estimates from our phase 2 to our phase 3 trials. So at Novartis, we have a lot of people who are really interested and excited about R Shiny, and they're building a lot of different applications in different programs and in different ways. Now, although this is really beneficial, it also poses some challenges. And one challenge is that the fact is that many people are developing Shiny apps in many different heterogeneous manners. Unfortunately, what that means is that when someone develops a really nice function, it's very hard to take that and apply it in a subsequent study. And so what we wanted to do is create a harmonized framework. And what this does is this really streamlines the app development. So my associate, Ardalan Mirshani, along with contractors from Rowe, there was Becca, Preston, and Natalia, really worked together to develop what we call the data pipeline. What we like to say is that this does the dirty work, so you don't have to. And what we've done is, let me go through the different tabs. Essentially what happens is we like to push this tab, which actually accesses our clinical data repository. So we have access to all of the underlying SAS data sets for a particular study. Then we can actually map the different columns into the charting modules. And what this does is this means that this can be done in a configuration file so that once the chart comes up, you don't have to specify the column that's required to make that chart. We can also configure subgroups and subpopulations, which are very popular functionality that people want. And then there's this abbreviation builder. Now, this actually has two functions. One of the things we recognize is that parameters actually have they have different levels associated with them, and R by default orders them by alphabet. And sometimes alphabetical order is not what you want, right? And so what the abbreviation builder does is it allows you to organize and give a specific order to that parameter level according to your desire, right? So you can actually pre-specify that. And then sometimes the labels for a different parameter are really quite long, right? So I can think of race and ethnicities tend to have really long labels associated with that. If you use those labels in your subsequent graphics, it's really going to negatively impact that graph. So what you want to do is be able to shorten down and abbreviate that label so that the resultant graphics actually display appropriately. What this harmonized framework does is it gives us consistency. And consistency means that then we can create modules that are plug and play. And to me, consistency is the key to scalability. It's getting better all the time, but we're not done. So we have some challenges that we're facing. So the first one is, how do we handle big data? So in our cardiovascular studies, these are studies that can have upwards of 12,000 patients. And the lab data is usually measured every month or so. And if you have 12,000 patients, 75 lab parameters being measured every month for a several-year study, this lab data is huge. In addition, we also have adverse event data from our large oncology studies. Another way data can get large is when we do our submissions. Sometimes when we explore when we look at data for submission, we have to pool several studies, right? And essentially what happens is that the app may work on those individual studies, but when you have to pool 7 or 8 studies together, well, things really break down. Then there's new data modalities such as imaging, digital sensors, and digital streaming information. And although this is only coming into Novartis a little bit right now, you can actually get the sense that it's growing, right? And so what we have to do is be able to handle all of these different data sizes. So I'm actually looking forward to the next presentation. So I had a chance to sit down with Viral Shah and Vijay to talk about the Julia package. And so here, the program here, and the idea is that not only can Julia potentially handle the big data size, that's only part of the problem. We also have to handle the computations, right? So this is something that I'm really interested to hear what they have to say in the next presentation. Then there's animations. You know, to me, animations actually are really resonating with our clinical colleagues, and we're being asked to develop animations even more frequently. So what I'd like to see is more packages to visualize temporal data or patient journeys. So I'd love to know what's out there and how we can apply different types of animations to our Shiny apps. I told you about the fact that we qualified our first Shiny app. Well, this is true, but it was quite challenging. I think the traditional qualification methods really break down when applied to Shiny apps. And what we really need to do is be able to streamline that qualification process. And finally, we have to increase awareness, right? Novartis is a big place. And although some teams have experienced the benefits, unfortunately, many have not. Right, so we have to increase awareness in education because honestly, seeing is believing. So in conclusion, we really need to focus on good graphical principles. And I presented to you the graphic principles cheat sheet here, which is free at this open source GitHub page. Now, the holidays are right around the corner. I can't think of a better gift than the graphic principles cheat sheet, right? You can give it to your family, you can give it to your friends, give it to your children. I'm sure they're going to love it. We also want to harness the potential of our data through graphics, but you can't use graphics alone. You have to use statistical thinking as well, because graphics alone can get you into trouble. Shiny is a powerful tool that really engages that clinical teams, right? We really want to promote learning from our data and reduce down those static tables and listings in favor of figures. I presented to you this harmonized framework, and what this allows us to do is to leverage each other's strengths and innovations. We want to see what works and then generalize that. You know, when teams come together and are highly engaged, with the addition of senior management support and analysts having fun, I know I'm having an absolute great time. That's the ultimate win-win. And with that, my daughter's tapping me on the shoulder and she's saying, you know, Dad, you still haven't answered the question, right? Would John, Paul, George, or Ringo have been famous if it were not for the Beatles? So on the one hand, you have a situation where you have each member of the Beatles being incredibly talented musician. And each member of the Beatles went on to have a successful solo career. But on the other hand, you have a band that together sold 600 million records and are often cited as one of the most influential bands in rock and roll history. So it's debatable. What is not debatable is that for me in drug development, I get by with a little help from my friends. And with that, I'd like to take a moment to thank those friends who've helped me get by, both internal and external folks, and especially this person in the lower right-hand corner, my daughter, who served as the inspiration for my presentation. So with that, Gugu Kuchub, everyone, thanks so much for your time. Really, really appreciate it. And one more thing before I finish, I'd like to say that for those of you who appreciate the Beatles, hopefully you noticed that I named or dropped 10 references to some of my most favorite Beatles songs throughout the presentation. Hopefully you caught them. For those of you who haven't, let me say, on the next slide, I'm going to show you where I actually made all of those references. So if you want to go back and listen to it, please shield your eyes. This is a spoiler alert. So here is the slide number, the song title, as well as the YouTube link to the song. Please download it. If you're not familiar with the Beatles, let this serve as an incredible introduction to an awesome band. So thanks very much, everyone.
Thank you, Douglas. That was an amazing talk. Big round of applause, as I saw in the comments. We have a few questions, and we do have time, I think, to take some questions. So the first one comes from Eric Nance, and it is, how was the Shiny app hosted and or distributed to the data monitoring committee via cloud deployment, for example?
That's a great question. So interestingly, so this actually had independent statisticians and an independent clinician at Novartis, right? So we actually had a mixture of both internal and external members that were part of the DMC. And so what happened was, right now this is hosted on our internal environment, and that internal independent statistician was able to share his screen, was able to log on and share his screen with the independent monitoring committee. So in a sense, the statistician drove the Shiny app for the independent monitoring committee and was able to display the information for them that way. The other challenge that we face is that we're going to have a situation where it's a fully external data monitoring committee, as I mentioned, and actually, you know, when you have to host this with the data security that's required for patient information, yeah, we're actually not sure how to do this. So, you know, I look forward to your ideas.
Okay, great. Another question comes from Gregory Stino. Can you describe steps to or highlight differences between qualify app and validate app, and whether is qualify equivalent to just fit for purpose?
Yeah, that's a really great question. I hate the word validation. I think it means too many different things to too many different people, and so I like to use the word qualified. I think qualified and validation actually, to me, mean the same thing. I just don't like the word validated. So to me, what we did is, you know, we wrote down the test scripts, as was mentioned in the talks earlier. You know, we mentioned several scripts, testing. We actually did the formal testing, the approval, and documentation. To me, validation is only maybe 10% about actually checking the code, and it's 90% about documentation.
Okay. Another question comes from Amrit Singh. How to plot visualizations with millions of data points and prevent lag?
Yeah, that's an excellent question, and that's something that we're working on. And so it depends on what the data are in order to do that. One of the ideas that we have is creating a database on the back end and only picking in the data that you want. Normally, you don't want to show it all at the same time. So for instance, if you have a scatter plot, right, if you have 10,000 or 1 million data points, it's just going to look like a mass of information. So it's not really going to be helpful. So we have to come up with different visualizations to make sense of that information. But what we want to do is you want to just pull in the information that you want, which can actually, you know, you can do that selection on the server side, and then bring into the UI only what you want to visualize, and that could be a way to look at that information. So, you know, for like lab parameters, you don't want to look at every single lab parameter simultaneously. You only want to look at one at a time. So that's a way to kind of cut down that information. The other way is to cut down the number of columns. You know, in our Atom data sets, they typically have like 140 columns. You don't need all of those columns. You only need like 10 or 12 of them. In a sense here, what you want to do is be able to only select those columns and use that, and that can also drastically reduce down the number and size of the data that you're reading in and displaying.
Okay, that's a double question coming from Eric Nance again. What efforts are being done to train your users on Shiny development, and how are the modules distributed?
Okay, that's a great question. So Eric, what we do is we actually have quite a nice, robust training program, and it's different levels of training as well. So we have an intro to Shiny course that's actually taught in different sections and modules around the different sites that we have. So that's been really successful. What we also have is intermediate training as well. We have what we call the App Hub, as we affectionately call it. And what this does is this is a repository to store those modules. And actually what we've done is we just actually created that data pipeline module that we've done before. And in this way, what this has done is this allows us to refresh all of those modules that we have available. And what people can do is download those individual modules into their application. And by storing them centrally, if there's any updates, we can actually update the central repository so that people always get the latest version of that module. So it's a nice way to keep track of where the modules are and to make sure people are always using the latest and greatest module.
Okay, great. Douglas, thank you again. That was amazing.
Hey, thanks everyone.